It's a great pleasure to have Nathan Babcock to talk to us on water as a quantum biomolecule. And Nathan's going to be introduced today by Alix Bailie. Now, Alix works as a Guy Foundation funded postdoc in Stan Botchway's lab at Harwell. And Alix, over to you to introduce Nathan.
It's my pleasure today to introduce this third episode in the topic on water. So this is the introduction to quantum effects of water associated with proteins. And today I'm introducing Dr. Nathan S. Babcock, who is a quantum physicist affiliated with Howard University in Washington, D.C. His research has spanned a range of topics in quantum information science and quantum biology for 20 years. He became interested in biomоlecular computing upon completing his undergraduate degree in physics at the University of Waterloo in Canada. He went on to do his PhD at the Institute for Quantum Science and Technology at the University of Calgary in Canada. He's worked on statistical modeling for structural biology as a postdoctoral research associate at Simon Fraser University in Canada, before pursuing research on the spin chemistry of aviation magnetoreception at the University of Exeter in the UK. Since then, he's worked on a series of groundbreaking studies investigating quantum mechanical superradiance in biomolecular architectures, ultra weak photon emissions, UPE, and the mitogenetic effect. He just released the first comprehensive treatise on quantum biology titled Physical Principles of Quantum Biology. His talk today will provide a concise overview of the quantum effects in water associated with proteins. Beginning with Einstein's seminal 1905 study on the quantum mechanical nature of warm, wet, and noisy matter, we will consider the fundamental role of quantum physics in determining the structure and chemical properties of liquid water. The importance of structural order in water between and around proteins will be examined in the context of biological electron transfer, proton tunneling, enzyme catalysis, van der Waals forces in DNA, and intrinsically disordered proteins, and ATP synthesis. The role of the water network as a mediator of quantum mechanical effects in biochemical reactions is proposed with implications for applications in medicine and biotechnology. Before I hand over to Nathan, I just wanted to give a very quick recap on just some background on proteins themselves. So, Frederick Engels said that life is the mode of action of proteins. And the way in which proteins are taught in schools is that we have our primary, our secondary, and our tertiary series of proteins. And these come together to form impressive structures that can undergo and undertake many different biological reactions. So while I've titled this slide People and Their Proteins, it could really be proteins and their people. As we sit here today, we are undergoing thousands of processes in many different cells in many different ways from many different proteins. These range from things like enzymes on the left here. Undergoing digestion of our lunch. We have things such as insulin controlling sugar in our bloodstream, controlling sugar. We have glucose transporters in our cell membranes. We have antibodies. We have our structural tissues like keratin, collagen, and elastin. And thinking that all of these are operating just from their sequence of proteins can seem a little bit ridiculous. So when we consider that actually it's the interactions with water that govern the folding, structure, stability, and activity of proteins, things start to make a lot more sense. So on the right here, what we have is the hydration shell that surrounds myoglobin. So like those protein structures on the previous slide, we just saw the protein structures, the twists and turns. But really, when we think of these proteins, we should think of them as being surrounded by a huge group of water molecules that they associate with and interplay with. This myoglobin on its own has nearly 2,000 water molecules associated with it that help it undergo its functions in the most optimal manner. 2,000 water molecules. There must be some fascinating processes undergoing that Nathan is going to explain to us shortly. So these water-protein interactions, they're not passive. The water is active in its relationship with our proteins and even the proteins in plants, such as those found in photosystem 2, which is well known for breaking down water to try and create energy and oxygen within plant cells. So an idea of proteins on their own really is past dated, and we need to be thinking about the interplay of these complex relationships with proteins and their waters. So it's my great pleasure now, without further ado, to hand over to Nathan Babcock today. Thank you.
Well, thank you so much for that very kind introduction. I could hardly ask for a better one. Of course, I'm Nathan, and I'm here to talk today about quantum effects in water associated with proteins. We'll discuss the importance of order and also, to some extent, the importance of disorder. You can see the funny sort of rectangular shape on the side of your screen is actually a pair of proteins you can see in a beige and a light blue color in a water box. This is the kind of structure that you would prepare if you're simulating proteins doing some kind of, one of their many physiological functions on a computer, of course, surrounded by, in fact, about 20,000 water shown here. So, without further ado, just to get everyone on the same page, we'll say a couple words about quantum physics. What is the quantum in quantum physics? It all comes down to this thing called Planck's constant, which tells you that the energy, say, of a photon, a particle of light, is proportional to its frequency, where the proportionality constant is h, Planck's constant. So our understanding of this aspect of quantum mechanics, the central aspect of quantum mechanics, goes back to work by Heinrich Hertz in the late 1800s, who showed in his experiments with the electromagnetic oscillators that if you produced a spark in one place, it could induce a secondary spark at a distance. And this was one of the first demonstrations of the reality of electromagnetic waves. But of course, not just any light would induce a spark in the second coil. It had to be the light from the first spark. And now, in fact, we realize today that this is because that first spark produces ultraviolet light at a higher frequency than visible light that we all see in the colors of the rainbow. And in fact, because the higher frequency carries more energy, it has the energy to induce a second spark, unlike photons of red or blue or green light. So this is one of the most elementary expressions of quantum mechanics in a simple experiment done over 100 years ago today. Now, today, we understand that quantum effects are inherent to every aspect of chemistry, and particularly for a somewhat more complex molecule like benzene with this cyclic aromatic structure. We see 6 carbons in black forming a hexagonal arrangement. It's very interesting to look at these systems because the close arrangement of the carbon atoms here allows the electronic wave functions, the electrons that hold the atoms together, to overlap. And this is the overlapping effect reflects a quantum superposition effect and also various entanglement effects due to the electron spin. So without getting too much into those details, what the take-home message from this slide is, as I've put in the quote here, that understanding complex chemical systems like biomolecules, as well as catalysts and novel materials, is a central goal of quantum simulations. So of course, looking at these quantum mechanical systems are a bit far beyond pen and paper or pencil and paper calculations that they might have done in Heinrich Hertz's time, and computer simulations are necessary to deal with the complexity of these systems. In fact, even a very simple molecule like ethane with just a pair of carbon atoms and some hydrogen atoms attached in white, to simulate truly from first principles, just the simple torsional motion of the ethane rotating around its main axis. If you really are simulating it from the core underlying quantum mechanics without any simplifying approximations or numbers put in by hand, it can take a whole room of computers like the Sunway computer I've shown here. So going to much more complex biomolecules, of course, is a real challenge. If we look at something like tryptophan, the amino acid that's reputed to make you sleepy after turkey dinner, you can see again these aromatic ring kind of shapes. And in fact, aromatic molecules like tryptophan and other aromatic amino acids are universal aromatic structure. And in fact, some work of my own and with colleagues cited here, we've shown that exciton transfer and evidently even superradiance, this quantum mechanically enhanced radiant effect that occurs when multiple photon emitters emit coherently in unison. Now, the main picture here, the larger one on the bottom right, is actually a picture of an amyloid fibril, the kind that's associated with Parkinson's disease. And what is interesting about this system is that these tryptophan residues are stacked up in helix, not unlike the pi-stacked helix that we see in DNA that we'll see on a future slide. So if we look at those tryptophans all stacked up, you can see them arranged here on the right-hand side. This is really a simple example in the most general sense of what we call coherence. So coherence is, broadly speaking, any definite phase relationship. What is a phase relationship? It's, if you have phases of the moon, or you look at the hand on a clock going around once every 12 hours. It's any cycle. So if you have a child and you're pushing them on the swing, of course, when you're on the swing, you want someone to push you just as you reach the top before you go for another swing forward. And, as any small child will tell you, if you push them at the wrong time, it's not going to be as effective for getting that, exhilarating height as you swing higher and higher on the swing. So even a child knows the importance of coherence. Daddy, push at just the right time. Don't get distracted. Now, coherence can be static, simply in terms of the arrangement of the pattern, either the cycles, the rings of each individual molecule, or, the repeating arrangement of the individual molecules in the helical structure themselves. It can also be dynamic in the form of oscillations, like clocks where the hands are synchronized, or the example with the child on the swing, pushing at the right time. Another case of a dynamic form of quantum correlations, coherence, is found in the van der Waals forces that I'll talk about a bit between molecules that are attractive forces between molecules that emerged as a result of synchronized fluctuations, rapid oscillations in the charge distributions of these things. Now, electron tunneling between different molecules like these are called cofactors, can also be synchronized in biology to enable efficient energy conversion. And tunneling, the ability for a quantum mechanical particle to not travel directly over a barrier, but to quantum mechanically surpass that barrier, is also an important effect that I'll discuss as we move forward, and crucial to many biological mechanisms. So one last word about these biological molecules. There's these things called coenzymes. And so these are small organic molecules, like a pair of tryptophan that have been bonded together to create the so-called tryptophan tryptophyllquinone, TTQ cofactor. So biological systems have wonderful ways of putting these molecules together in ever more complex forms. And the structures themselves give them interesting, as I mentioned before, photophysical, photochemical, and electron transfer properties. So TTQ, in fact, if you look at the figure in the upper right, is an electron carrying molecule, a molecule which transfers an electron from an enzyme at the top, MADH enzyme, during the digestive processes of soil bacteria. The red protein transfers the electron to the blue protein below it, the amocyanin protein it's called, and it's transferred from the TTQ cofactor to a copper atom, actually. But of course, it has to get from one protein to the other, which means crossing the gap filled with water between the two proteins. So that's basically the intro to quantum mechanics as it applies to biology. Where does the water come in? Of course, as Alix mentioned, the proteins are constantly surrounded, bathed in water. There's this ordered hydration layer around each protein. And if it needs to transfer an electron or carry out some kind of docking mechanism with another protein, of course, the water is there and is involved in the process. Now, water itself is warm, wet, and noisy. This is in part due to a quantum effect known as Brownian motion. Brownian motion is also a thermal effect, but what's quantum mechanical about it was that Einstein, in his seminal paper on this in 1905, was ever to show that certain features of the Brownian movement were characterized by the underlying atomic structure of matter. And of course, the existence of atoms themselves, quanta of matter, is one of the most fundamental aspects of quantum mechanics that at that time was still under dispute. And it wasn't until the experimentalist Jean-Baptiste Perrin went on to verify Einstein's theory of Brownian motion experimentally, in effect demonstrating the existence of atoms, that many, this is what it took ultimately to convince some of the last holdouts against the atomic theory of matter that in fact atoms were real. And of course, Perrin won the 1926 Nobel Prize in Physics for this work. So I hate when people say the structure of matter itself is somehow trivial quantum mechanically. 100 years ago, this wasn't a trivial concept at all. But it only gets more complicated from there. The electronic structure of liquid water is itself highly complex. If you look into the literature, I discovered recently that actually we didn't have simulations to predict that ice floats until about 10 years ago. In fact, there's complex quantum mechanical effects, again, inherent to these so-called van der Waals interactions and so forth, that need to be fully accounted for, even to predict accurately the fact that ice floats and doesn't sink when you put ice cubes into a glass of water. Even more recently, we have been able, with computer simulations, to finally predict using very advanced, what are called many-body perturbation theory calculations, to predict the ionization potential of water, the electron affinity of water, the band gap of water, which is important if you're a materials physicist or you're interested in things like semiconductors. But something like the ionization potential, which is the energetic cost for the system to release an electron, the cost of an energy of a cost and energy of an electron escaping, something that we touch on in high school physics, but was in fact not able to be calculated from first principles until the last few years. So the underlying quantum mechanics, even of liquid water, cannot be understated. Now, how do we simulate this? Often in simulations, when we're looking at the actual functional characteristics of proteins, we don't necessarily need that level of electrochemical detail, and effective classical simulations will suffice. So in the picture here, we have a pair of proteins surrounded by about 20,000 water molecules. And in general, these water molecules are important for a number of functional aspects of these proteins. They can be involved in transferring electrons between the proteins. They stabilize protein conformations. In fact, protein folding is itself driven by the hydrophobic nature of the protein. The water molecules are important often at the active sites of enzymes that carry out biochemical transformations. They can mediate protein interactions. They can facilitate proton tunneling. And I'll touch on the so-called Grotius mechanism later on in the talk. And of course, they're involved in protein dynamics. And as I mentioned, protein folding, and as we'll see, also DNA repair. So there's really no aspect of, protein function that doesn't involve water. If we look first at all at electron transfer between proteins, this is the topic of which I have the most experience. Again, if we look at that TTQ molecule that I showed on the previous slide, shown in purple, and the copper atom, also shown in purple, connected by a kind of a blue zigzagged pathway. This is, according to our simulations, the electron transfer pathway that the electron takes from one coenzyme to the other, from one protein to the other through a matrix of water that sits between the proteins. And in our studies, numerically, we found good evidence to support the idea that actually the protein has a fairly, somewhat sophisticated mechanism for controlling the water that sits between the two when the actual electron transfer reaction occurs. So to understand a bit more about electron transfer theory, if we look at another electron transfer system, this is a pair of heme molecules that gives color to your blood. It's an iron atom in each heme surrounded by an organic lattice that holds the atom there. An electron can be transferred from one heme to the other through an arrangement of, if you can see, three organic molecules that effectively form a kind of bridge-like system between the two heme cofactors. So that when the electron transfer occurs, in fact, the so-called coupling matrix element, this TDA, is a quantum mechanical term that we use in our calculations to estimate the strength of the coupling between the two hemes. The strength of the coupling that is necessary to transfer the electron is much weaker if the electron has to travel through space without any intervening bridge molecules. But if we put a nice electron-rich molecule or several in between as a bridge, the electron can be transferred more efficiently between the two using an effect known as superexchange, which is, again, a fundamental quantum mechanical effect that is related to the actual spin coupling between the electrons. For any physicists in the audience, this is Anderson-Kramer superexchange. Pardon me, Anderson-Kramer superexchange. So we know about electron transfer thanks to the Nobel Prize winning work of Rudy Marcus. Beginning in the 50s, he began developing his theory of electron transfer, and ultimately, culminating with, very comprehensive quantum mechanical expression of the theory in the 1980s, for which he won the Nobel Prize in the 1990s. Now, in that picture of electron transfer, you have two surfaces, a reactant surface and a product surface. Now, we usually think of these in terms of the electronic energy potential surface in which all the nuclei sit. So if you imagine all of the nuclei of the atoms, oxygen, hydrogen, and carbon atoms in your system, they're surrounded by kind of like an electron cloud that provides the potential, which ultimately gives structure to these molecules. And if an electron transfers from one molecule to another molecule, the shape of that potential changes. And so you actually have two potential surfaces, the so-called reactive surface when the electron is at the molecule donating the electron, and the surface for the system after the electron has been transferred to the molecule receiving or accepting the electron. Once again, that electron transfer coupling is governed by the so-called coupling matrix element, which involves a number of Hamiltonian terms and energy differences in the system in order to calculate. But ultimately, the important thing to understand here is that the system, the nuclei have to travel along the reaction coordinate to the crossing point between the two potential surfaces. And this only occurs sporadically, intermittently in the system. And it's further complicated by the fact that actually we need to think of not just the energy potential surface, of the electron cloud in which the nuclei sit, but what's called the free energy surface, which accounts for thermodynamic aspects of the system as well. What happens when the electron reaches the crossing point is that it can effectively undergo a quantum jump. As Erwin Schrödinger said, if all this quantum jumping were really here to stay, I should be sorry I ever got involved with quantum theory. But it is here to stay, and these quantum jumps, these tunneling mediated transitions from the donor state to the acceptor state of the system really are crucial to understanding simple electron transfer reactions in biology. Now, some of you may be wondering, Dr. Babcock, what has this got to do with water? Well, the crucial thing in Marcus's reasoning, the thing that really got him his Nobel Prize, was understanding the role of water in these systems. I mentioned that ultimately it's not simply a potential energy surface, but a free energy surface. And what this means is that actually we're waiting as the system samples all the different arrangements that it can explore, in thermodynamic phase space, as we call it, all the different wiggles and jiggles that it can make. It needs to wait till just the right moment. And so if you look at the cover of one of my favorite textbooks, Chemical Dynamics and Condensed Phases by Nitzan, in fact, this is the diagrammatic picture of the reasoning that Marcus went through in order to win his Nobel Prize. So we have in the top left, top right, bottom left, and bottom right pictures of a pair of charge-carrying systems. In physics, we often use the letter Q to represent charge. And so if you imagine a charge sitting at a molecule or even a little metal sphere, an electrode in water, that when the charge is at one sphere, if we look at the top left picture with the pair of spheres surrounded by they look like little pills. They're meant to represent water molecules. That in fact, the sphere with the Q equals 1 shows all the little pills lined up with the white side facing the sphere. And the sphere without the charge at it, where it says Q equals 0, the molecules, which are again intended to represent molecules, water molecules, are completely disordered. Now, the electron could just hop from the right sphere to the left sphere, according to the blue arrow that's crossed out. But the blue arrow is crossed out for a reason. And if you look to the top right figure with the two spheres, now we see that indeed the Q equals 0 on the right, the Q equals 1 on the left, but something has happened. In order for the charge to move from one sphere to the other, and for the system to relax thermodynamically, the water molecules on the uncharged sphere will now become disordered, while the water molecules on the charge around the charged sphere, around the charged sphere are oriented. Because of course, water is polar. And the positive side of a water molecule will face the negatively charged body. Now, in order for this to happen, there has to be a complete reorganization of the solvent, the so-called solvent reorganization in Marcus theory. And in order for this reorganization to occur, it doesn't happen for free. Ultimately, there are steps involved. If the charge just hopped suddenly in this quantum jump from the one sphere to the other, without the solvent reorganizing, that would revolve a lot of energy, because now all of a sudden the charge has transferred from one sphere to the next without the solvent having time to relax, and the solvent is now in a higher energy state, because wherever the charge is, the solvent is going to rearrange in order to find the minimum energy configuration where all the positive and negative charges line up. So actually, Marcus reasoned that, due to this effect, in fact, due to conservation of energy, that the electron transfer could never occur directly. That in fact, if we now look at the red arrow labeled 1, that what must happen first is the system has to wait until, based on the wiggles and jiggles, the Brownian motion and so forth of these systems, that there is a fluctuation where now, in fact, all of the water molecules are more or less disordered around both spheres. Now, you have to put energy into the system. The system would have to, absorb a heat fluctuation, absorb a little bit of energy in order for this first step to occur. But now that the system is symmetrical, if you look at the bottom two pictures, there's no difference whether the Q equals 1 is on the right sphere or the left sphere, because the solvent is completely disordered in each case. So now it has a chance to hop from one sphere to the next, which occurs in step 2, the little blue arrow on the bottom. And in fact, once the charge has hopped from the right sphere to the left sphere in this quantum jump, now the system can relax into the final picture that we have on the top right, where the water molecules, the little pills in the image, have reoriented, all facing the white side of them towards the sphere. And these are the necessary steps that are in fact shown when we go back to our Marcus theory picture. It's that reorganization of the solvent that is the actual step occurring as we go to the crossing point of the picture. And as it now goes to the bottom of the product surface, this is the relaxation of the water as they now orient towards the charge acceptor. And so it's very interesting to me, when I studied electron transfer, during my PhD, to learn how much the solvent really actually controls this electron transfer step. And Marcus's genius was just recognizing that energy conservation would be violated if the electron hopped from the donor to the acceptor without the water molecules being able to reorganize as well. And therefore had to involve this multi-step process, which actually creates a free energy barrier, which resists the electron transfer, prevents it from happening spontaneously in most cases. So we studied this kind of effect in the proteins, the methylamine dehydrogenase, MADH, and amethionine. And we wanted to see exactly what was going on. But the water is involved more than just, the order or disorder of the water in terms of controlling the solvent reorganization. If we look, again, I mentioned that there's actually a few water molecules sitting between the proteins if you look closely at the kind of blue pathway between these two. And in fact, if we look at the surfaces of the proteins, I'm showing the MADH surface on the top on the right and the amethionine surface on the bottom, where the different charges on the proteins are shown in white, red, blue, and yellow. In fact, we have this hydrophobic region in white with kind of a dimple on it on the amethionine. And that connects to an indentation on the MADH where it can form a seal in order to collect just a small number of individual water molecules in between the two proteins while keeping the bulk water out. Now, if we look at that with just the donor molecule, the TTQ, and the acceptor molecule, the copper atom, surrounded by the molecules that coordinate its placement in the enzyme, you can see only 1, 2, 3, 4 water molecules sitting in the center here. And we have hydrogen bonds. In fact, a hydrogen bonded network shown with the dotted lines in this system. And now in this case, in fact, the water molecules are forming a bridge to allow the electron to cross. And so, in fact, the water for something like an electron transfer reaction do not just control the opportunities the system has for the electron to hop due to thermodynamic reasons, but actually provide the bridging effect, that quantum mechanical coupling term that I mentioned as well. And so we performed a number of molecular dynamic simulations. These were classical molecular dynamic simulations, and we found a number of different pathways. We had kind of a class we called the A pathways, from the TTQ to the copper at the bottom, where they were well bridged with nice arrangement of 1 or 2 water molecules in between. But then we had kind of a B grade pathway, which was not as well connected, a C grade pathway, and a D grade pathway, where you can see actually these solid ovals represent through space jumps. It's not completely connected. And of course, with the fluctuations of the system, in our simulations, as water molecules jiggle around and bounce around, the question was really what kind of systems would produce, the most robust bridging effect? When was the bridge there, the most consistently? We were motivated to do this work based on experiments where they performed mutations in the laboratory on these proteins. They took a single amino acid residue, in this case a methionine residue, which you look on the left-hand side, you can see the methionine 51, which sits on the amethionine, and removed it, replaced it with a leucine, lysine, or an alanine amino acid instead. And they found, if you look at the right figure with the K3 as the electron transfer rate measured in their experiment versus temperature, they could see that in all cases of the mutation, the electron transfer rate went down. And in fact, in the case of the methionine to alanine, M51A mutation, the electron transfer rate decreased dramatically. And they speculated that it could have something to do with the water molecule, which had been revealed in previous crystal structures dating back to the 90s. Now, in our simulations, we found that indeed, if we looked at the so-called wild type simulation with the methionine residue there, we had only about 2 or maybe 3 at most water molecules, about 2 molecules on average in the ET region, that protected region between the 2 proteins. But when we when we changed to leucine, lysine, or alanine at that same 51 position, then in fact the number of water molecules sitting between the protein increased as many as 3-fold. And in fact, we found, based on these different mutants, if you look at these figures now, we have a wild type in the second panel, the methionine to leucine, the third panel, methionine to lysine, that's panel C, methionine to alanine, and methionine to an additional one we looked at, cysteine. In fact, in all of these cases, if you look at the probability of a certain pathway, like the preferred A1 pathway, occurring, as we sample all the different molecular conformations in our simulations, the term characterizing the electron transfer element, in this case we show it as E total, that it was the largest, greater than 1, in the wild type system, and that all the other pathways, A2, A3, B, C, D, and E, all of these other pathways had greatly reduced coupling. You can see that E total goes down to, less than half in some of these. And in fact, this really showed us that the mutants were no longer able to consistently create that bridged water molecule between the 2. And when we looked closely at the simulations, we could see indeed that methionine residue in the original native protein, the wild type protein, was a barrier keeping out the bulk solvent molecules, the water molecules. And that when that particular amino acid residue was replaced with something else, it no longer had just the right structure to keep the water molecules out, and the more bulk-like, water molecules, you can see them almost lining up to get into the region. And it's interesting because you might think naively that more water molecules would, offer more bridging opportunities between the 2. But actually, it was the order of just 1 or 2 water molecules that could find exactly that nice hydrogen bonded position in the native system that allowed the more ideal electron transfer to occur. And so we called this molecular breakwater, this methionine 51 residue working in concert with that other molecules, the other amino acids in that puckered region around the amosine and active site. So I hope this hasn't been, too much very specific information, but the idea I wanted to convey to you is, the degree to which water molecules control, in fact, seems almost every aspect of the electron transfer in this kind of system. So, unless there's any questions at this stage, I will move on to the more general portion of the talk to discuss liquid water and its role in a number of other protein and DNA related effects. Great. So how do we simulate water? As I mentioned, usually, for a lot of our purposes, simple classical simulations are enough. And so one of the most commonly used water molecules is the so-called TIP3P water molecule. And this is I believe it's the transition interaction potential. And the 3P stands for a 3-point potential. You'd have to check me on that acronym. But the bottom line is that in the TIP3P model, the two H's have a plus 0.4 charge on them, and the oxygen has a minus 0.8. If you remember from your introductory chemistry, indeed the water molecule is a polar molecule with a negative charge on the oxygen and positive charge on the hydrogen. Now, there's also a little more sophisticated 4-point potential in which the hydrogen and oxygen have an additional, it's like an imaginary atom placed just below the oxygen. And now the charge is on the M, but this is just a charged part of the system, whereas the mass is still on the oxygen. And indeed, this 4-point potential can be better for some simulations, but for the vast majority of purposes, TIP3P is optimal. Now, for some things, the typical way that this is parameterized, in fact, the results of simulations are poor. So if we look at not well-folded protein, but what's called an intrinsically disordered protein that doesn't fold up into a well-defined optimal structure, that in fact, the TIP3P model can produce a very poor performance. In fact, it will predict that the intrinsically disordered protein folds into a globular structure. But we know in real life that not all proteins have final well-folded state. It seems that the TIP3P model was parameterized in order to get proteins to fold or stay folded in simulations, but was not adequate, when it was originally parameterized. In fact, people weren't often thinking about intrinsically disordered proteins. And the understanding is that the effect of what's called the dispersion in a system, this is a form of van der Waals force that's due to very rapid quantum mechanical fluctuations in the system that produce an effective protractive potential, is necessary in order to fully account for these intrinsically disordered proteins. And what another group working with D.E. Shaw, if you're familiar with that group out of New York, they have some of the fastest simulations for these kind of systems in the world. They introduced a modified set of Leonard-Jones terms. The Leonard-Jones terms being the specific interaction parameters that are used to account for these van der Waals forces between molecules. And so choosing enhanced van der Waals term and reparameterizing the TIP4P model, they were able to get dispersion that actually reproduces the reasonable disarray, the reasonable, I should say, lack of structure in the protein as it remains in its unfolded state. Now, these still aren't polarizable water molecules. Water molecule can wiggle and jiggle, and a polarizable water molecule, as I've shown here, the SWM4, SWM stands for simple water molecule, and the DP corresponds to the polarizability. In this case, it's actually a Drude oscillator. The D stands for Drude, where we now have a charge on a spring in the system, bond with a force constant, that allows the actual degree of polarization, the dipole moment of the water molecule, to change. Now, there's more sophisticated versions of that. An amoeba force field, which stands for atomic multipole optimized energetics for biomolecular applications, where they include a number of parameters on each molecule to fully account for its polarizability before you get to an ab initio quantum mechanical model of a water molecule, and not unlike that ab initio picture of the ethane molecule I showed, I think, on my third slide. And these are typically done with density functional theory in practice, DFT. If we want to understand water-mediated protein interactions, it all comes down to these hydrogen bonds. So I've shown here a picture from this reference in the Royal Society's Chemical Biology, that the hydrogen bond, of course, forms between the positively charged hydrogen and the negatively charged oxygen in a separate water molecule. Even liquid water tends to be fairly well ordered, although it's dynamic, because the hydrogen bonds, of course, are responsible for many of the unique properties of water, the high degree of surface tension, for example. Now, when water sits near protein or DNA, the positive hydrogens can form these weak hydrogen bonds with oxygen or other negatively charged atoms on the protein surface. And this is what creates the hydration layer that Alix kindly introduced in her talk. Now, the hydrogen bond can break and reform with another water molecule, for example. But in order for the protein to undergo any conformational change or any, significant change in its shape during protein folding example, all these hydrogen bonds will constantly be forming and reforming. So the solvent really controls a great deal of the protein conformational sampling and ultimately its dynamics during a relaxation process, for example. If we look at the degree of water-mediated protein interactions and the timescales involved in these, we see a lot of different things going on. At the shortest timescale, we just see simple hydrogen bond vibrations, just wiggling back and forth. Once you get to 1 to 10 picoseconds, we can have hydrogen bond breaking and rotational relaxation, as well as translational diffusion. And at the 10 to 100 picosecond time scale, we can see collective water dipole relaxation. Now, this is the kind of thing you might especially want a polarizable water model in your simulations in order to actually observe dipole relaxation effects. Now, as you go to longer timescales on the order of a few nanoseconds, you may see side chain fluctuations in your protein because, of course, even for the side chain to move around, all the water molecules have to reorganize in effect. It's not unlike the solvent reorganization that I showed is necessary to enable an electron transfer process to occur. Protein tumbling is an order of magnitude longer, and finally, full-scale conformational transitions, allosteric transformations in a protein, for example, might take an order of microseconds. So Philip Ball, in Proceeding of the National Academy of Science article, wrote that low-frequency, large amplitude modes in the terahertz range are particularly important in controlling the conformational changes that dominate protein function. However, at this time, there's no simple qualitative account of how protein and solvent dynamics interact. So we know to a great degree the nature of the interactions and the types of different movements that we see, but because of the high complexity of these systems, the involvement of van der Waals and polarization forces, there's just so much going on in so many timescales that it's very challenging to predict in advance what is really necessary in a simulation without going to a full-scale ab initio approach. When we look into cells, of course, these kind of hydration layers are everywhere. covering every part of the system. If you zoom into a globular protein, everywhere you look, you see these biointerfacial water molecules that are separated from the more bulk-like water. But in fact, even in a relatively simple bacterial cell, studies have shown that the solvent, the cytoplasmic fluid, actually has a kind of glassy character, and that it's not even necessarily well simulated as simple bulk fluid. Again, even in a fairly simple cell like a bacteria, let alone in a eukaryotic cell, which is rich in structure all throughout the cell. If we look at DNA, the hydration layers are also extremely important at the level of, a distance of about 18 angstroms away from the double helix of the DNA, which is almost 2 nanometers. And they call this hydration spine that wraps around the DNA and has an important role in controlling its properties as well. Now in DNA, the electrostatic repulsion between the stacked base pairs is offset by van der Waals forces. These are these attractive forces due to rapid quantum mechanical fluctuations. And the fact that quantum van der Waals forces allow structural stability as well as, it's believed, wave-like electron transfer through the DNA. Now, the hydration bond dynamics and water reorientation times, again, range from picoseconds to a few nanoseconds in the hydration shell around the DNA. And these hydration layers are crucial to DNA structure and function. And I've provided a couple references here that I can recommend if you want to look into these kind of characteristics of DNA hydration shell in more depth. Now, the importance of the hydration shell around DNA proteins have led some, like Philip Curian at the Quantum Biology Lab, to propose that the electron-electron correlations are mediated by photons in quantum field theory, and analogously, long-range correlations in the molecular water field between DNA and enzymes may be mediated by dipole waves. Now, we already, in fact, know to some degree this is true because we know that van der Waals forces, these, rapid, concerted, correlated quantum fluctuations are important in understanding the structure of water. And if you want to learn about, more in-depth studies of these many body interactions in solvent protein systems, I highly recommend the Science Advances article from 2019 that's cited at the bottom of this slide as well. So if we now look inside an enzyme again, we can look, for example, at the operation of base excision repair by the enzyme MUT-Y. So this is a DNA repair enzyme that is able to come in and cut out aromatic base from the DNA so that it can be replaced later on. Now, in this case, if we look at the figure where we see now the nucleotide base has been pulled out of the helix, and we zoom into that, we can see an arrangement of 1, 2, 3, 4, 5 water molecules that sit at the active site of the enzyme. And in fact, these arrange a concerted proton transfer effect where a proton is transferred through the water network to the nucleotide base, here coming into N7, and at the C1 position, a water molecule is able to come in, break the bond between the carbon and the nucleotide base and form a hydroxide at that carbon site instead. And so, in fact, the water is actually intrinsically involved in the cleavage reaction here that snips the nucleotide out of the DNA as part of the overall repair process. If we look at another system like the oxygen evolving complex that's found in photosystem 2, involved in photosynthesis, that this work published in JICS in 2021 has shown that the actual proton sitting, in this case, at the W1 site of the enzyme active site. You can see it in the light blue circles here. There's W1, W2, W3, W4. These are all actually water molecules that have become part of the active site. These are water molecules that have prosthetically bonded into the active site itself. And to me, it's just incredible that, you know, the water is not actually just involved in enzyme catalysis. It actually becomes part of the enzyme proper. Now, and in this case, at the W1 site, you have the water molecule bound into the enzyme. We have another hydroxyl group, that's an oxygen and a hydrogen at the D61 site. And in fact, the hydrogen atom will tunnel back and forth between the W1 and the D61. And if you see it on the picture on the left, you know, this is the simple kind of cartoon image they've shown to show that the quantum proton ultimately, through this back and forth tunneling process, forms a coherent superposition of sitting at both sites. And the importance of the proton delocalization enzyme in this system appears to be essential for enabling electron transfer, for disabling proton transfer. It keeps the proton from escaping during the enzyme process. It's important for the accumulation of oxidants that are necessary in order to actually carry out the enzymatic reaction, and also plays into an isomerization step in this system. So you can really see, you know, the involvement of water, proton tunneling, and so on and so forth. They all seem to be very essential in the oxygen evolving complex that ultimately takes water and produces oxygen during the photosynthetic reaction we all learned, where carbon dioxide and water form sugar and oxygen gas during photosynthesis. Here's the close-up where we can actually see, if you see the little R primed OH and ROH, the arrows show the dotted line where the proton is actually tunneling back and forth between the D61 site and the W1 site. So finally, just before I conclude, I wanted to say a couple words about the actual pardon me, this is the ATP synthase mechanism that's found in mitochondria. So if we look at complex 1, complex 2, complex 3, the usual picture of the system, we have the pumps that pump hydrogen across the membrane in order to drive ATP synthase in the system. Now, in this case, typically we have a picture of the so-called Gratis mechanism, where we have a proton wire that enables protons to traverse the membrane, usually for a proton to cross a membrane. Well, in some pictures, the membrane is completely resistant to protons crossing it, but others have speculated that the membrane may be permeable. But in order to, you know, bring the protons across in a controlled way, we have these OH groups either as part of the membrane or a water channel that allows the proton to cross through a Gratis mechanism, where the the proton hops from one water molecule to the next. But it's been speculated, in fact, that complex 1 may in fact involve Brownian motion proton current, where the actual protons travel through the membrane and then have a Grotus type mechanism, proton hopping mechanism, like a bucket brigade, that drives the ATP synthase. So you can read about that in Morelli's Open Biology, and also see another related view in Frasch's Frontiers of Microbiology paper 2022. So in closing, despite some views to the contrary, biological systems are incredibly complex with quantum effects. And you can, I hope, see that even our understanding of the water molecules in the system, often we have a whole level of approximations from simple classical models to extraordinary complex quantum mechanical models that are needed to actually account for sometimes even relatively simple chemical properties like the ionization potential of water. And in order to go to something like an enzyme active site, we need high performance architectures, quantum computing, or in some cases, we've had new parameterized force fields being emerging from machine learning models. So often hybrid models are also required that include both classical and quantum parts of the system. In conclusion, water is fundamentally important to all biological process. Water mediates many quantum mechanical effects in physiology, and the quantum mechanical structure of water itself is also essential. So I think I couldn't agree more with Einstein when I say that one can best feel in dealing with living things how primitive physics still is. This was Einstein's view. He wrote in a letter to Leo Szilard. And I invite you to check out my new book, The Physical Principles of Quantum Biology, on the archive. You can take a screenshot of that or just Google it. And finally, I'm happy to take any questions that you may have. Thank you so much.